you. So this is, I believe in giving talks that are entertaining, not necessarily useful. So that's been my mission. So let me start out by saying a few things. So in the last couple of years, as everybody knows, deep learning has been taking off. At Argonne, we have a couple hundred people that keep coming to various meetings and workshops that are engaged in deep learning, and some subset of these I hope to get interested in neuromorphic computing. A couple of months ago, we started engaging with, we've been talking to Intel for a couple of years about the project that they're doing with Mike and his team. But then there was the announcement at CES, and a few weeks later, we got a little test machine. And so we're in the process of setting up a test bed. And I think Mike will talk, well, he's already talked about it some, he'll talk more. And of course, there's the Wiki Day on Friday. So one of the things that we're doing is we're setting up a test bed. And one of the reasons we're doing this, of course, is we have a large collaboration with Intel on the exascale project. And this is in the spirit of continuing engagement. So we have a plan to do a number of open source tools on this hardware to extend the programming environment that Intel's building, to stand up some exemplar applications that are different from the ones that Intel's been working on, and put together some training and courses on this. Ian Foster and myself have committed to teaching a 2-quarter series starting this fall at Chicago that is designed to connect to each other, one on learning systems and the second one on neuromorphic computing. That should say fall, that should say spring of 2019 on the second one. They're not overlapping. They're one after another. So we have a team that's spinning up. I'm going to do a walkthrough of a number of applications. I'm not going to talk through all of these because it's too much, but I'm going to try to talk about the first 5. So they're kind of bridge the space between edge computing examples like the urban sensing, some, I would say, and the real-time analysis of biological samples is also an edge computing kind of example. And then the cephalopod neuroscience, computational neuroscience example, and the mammal brain circuits, those are kind of more computational neuroscience. And then I'll talk about autonomous prospecting, which is my personal favorite that I'm pushing. We have many other projects. There's about 60 projects at the lab that are engaging in some form of aggressive machine learning, many of them in deep learning, and some subset of those will, I think, be targets for neuromorphic. We're still trying to understand the degree at which hardware like this is more of a test bed to experiment with ideas versus something that would be practical on real problems. But work that we have going on that in some cases very large scale that I have slides in here, but I won't have time to go through them. We have a project with high energy physics in the Large Synoptic Sky Survey Telescope that's building a system to apply real-time processing of streaming data to look for gravitational lensing objects. We have a very substantial project in cancer and drug development that's using various types of machine learning, and we'll tinker with that in the context of neuromorphic. We have a very aggressive group in materials design, and one of the things that we intend to do over the next couple of years is close the loop between systems that can monitor and learn things about other systems, and I call that, in this case, autonomous computing, where we would ideally be able to build a system that uses neuromorphic computing to keep track of our very large-scale system, but I won't probably have a time to talk about that. So let me talk about the array of things. How many people have heard about this, at least a little bit, because it's been in the news? Okay, so this is a project that's supported by the lab. It's a partnership with the City of Chicago, the University of Chicago. NSF has invested a large amount. Laboratories have invested a large amount. The basic idea is to stand up an array of sensors, in this case about 500 sensing platforms. I'll walk through what they are, around the city of Chicago. And these sensor platforms have cameras and microphones and trace gas sensors and other types of sensors in them. And we've been building an architecture to support this. And one of the challenges is pushing real-time inferencing and to some degree online learning out at the edge. So the canonical device is a stack, platform stack that attaches to light poles. And so we got 500 of these are going up, blankets the city, and we have a cellular network backhaul for free. The city's public works people put them up. We gave them party hats. They're really happy to do this. And so what we're doing is we're dramatically increasing the density of sensors in the city. So this picture on the left here is like how many sensors are there because of, say, the EPA or something like that. Our mesh, of course, is like, imagine 500 things to sort of saturating that. Most of them are at intersections, and they have cameras that look both up and down. Up so we can see the sky and interpret what's happening, say, from weather, precipitation, clouds, and things like that. Others are pointed at the intersection. This is, of course, a cartoon. But the idea is to be able to characterize what's happening there, look for near-miss accidents with cars, pedestrians, try to capture things. Here's a snapshot of one of our existing, real data, right? So it gives you a sense of what the field of view of the camera. You can see the snow on the ground. It was from a couple of days ago, about 2 weeks ago, actually. And we have pretty conventional computer vision systems deployed. There's a small GPU accelerator on the box. And it's because of the privacy issues, we're doing all the inference in the sensor, and we only ship back to the mothership interesting events and statistics, and we pledge not to record and track people from one sensor to another. So we're trying to walk the line between interesting science and privacy. This thing is actually being stood up as a research platform. So we have dozens of research groups around the country that are able to use it for whatever they're interested in doing. One of the side goals of this project is to push DOE away from traditional sensor environments where you would spend lots of money on highly calibrated, sparse sensor arrays towards thousands of lower cost but calibrated populations of sensors, and be able to, again, put the intelligence in the edge. There's just the team that's working on this, Charlie Catlett and Pete Beckman and others, never thought they would get into kind of low volume manufacturing, and since they've kind of contracted all of this out. But this shows the inners of the platform. It's very robust. It's designed to live on a street pole. It's not heated or anything, because it generates a fair amount of internal heat. It's completely remote controlled, so there's two complete separate operating systems, one that's running the kind of analytics and the data collection, and one that's just a mothership, that is kind of the control node, kind of a phone home node that never is very, very robust and can reboot the system and takes care of it. You can see the higher, I don't have a pointer, but on the bottom you can see the stack that's got some compute power on there. The software environment for this is called Waggle. This project's been very aggressive in partnering with groups around the world, and so there's many, many groups that are starting to deploy these nodes. And one of the goals we have for the partnership with Intel and the Neuromorphic project is to understand how much improvement in power and the kind of variety of inferencing tasks that we might do out in the field in this infrastructure. So that is a target of this program. And there's a cool movie showing somebody hand building a little bit faster than real time. After you build about a dozen of these, you never want to build anything anymore, so it's all contracted out. And they partnered with the Art Institute in Chicago to make a nice design, and lots of students are involved in that. Okay, so that's number one, pushing these devices out into an urban sensing environment. We get a stream of data back to the lab that's completely open. Lots of researchers connect to that stream. The connection with Neuromorphic is to imagine how we might miniaturize this, make it much more low power. This thing is plugged into wall power because it's on a light post, right? It's not batteries or anything, but we're very interested in miniaturizing this and understand how much inferencing we could do with a very low power continuous device. All right, second example is real-time analysis of biological samples. For the last 15 years, my group, including Fang Fang, who's here in the audience, is going to be here a week, we've been working with NIH and the National Institute for Allergies and Infectious Disease to run a bioinformatics research center that grew out of the post-9/11 biosecurity uptick in the country. And over the last couple of years, we've been applying machine learning to predict things like antibiotic resistance in literally hundreds of thousands of genomes. And what's interesting about this problem is where it can go in the very near future. So the historical model here is you go out in the field, you collect samples, you somehow deliver them to your lab, you process them on a DNA sequencer, and then you have some data, and then you use a kind of grounded infrastructure to process that to make some inferences about it. And of course, whether it's for disease or environmental queries, the challenge is the distance and the time to do that. With some new technology like the Oxford Nanopore device, there's a small chip-level device that can be brought out into the field where you can actually collect the sample and do the DNA sequencing while you're in the field. So we're interested in that trajectory. So this is a little device. It's a little bit bigger than those of you that have like an Amazon Fire TV, it's almost exactly the same size as a Fire TV or like, a jumbo USB drive. And this thing has got a multiple use cartridge in there that uses nanopores embedded in a membrane to measure ion currents to do the sequencing. And the cool thing about it is that it's portable. So you could drag this out into the field. This is a group that was using it out in the Arctic. And you plug it into a laptop. Of course, it wants to talk to, the way the software is designed, it wants to talk to the mothership to interpret the data stream coming off of the chip. If you're going to do it really out in the wild, you have to break that link and make all the software local. But the thing is designed so that you minimize sample prep, and you can, if you know what you're doing, in a few minutes you can put a sample in there and you can be sequencing. And of course, when it is producing continuous data, and you can stop when you have enough data. Now, there's two things that are interesting about this. On the left is the molecular pore that it's pulling a single-stranded DNA through, and it generates this kind of data on the top that's kind of stochastic data. And that has to be interpreted to generate the DNA sequence. So the first problem is how well we might be able to use a device like a neuromorphic chip to actually do this in the embedded sense of the sensor. So that's challenge number one, and that they've published a lot of code for doing that. The second thing is that sample prep, even though it's relatively straightforward, you're still doing it manually. Oxford Nanopore bought a company called Voltrax, which is making another chip that uses magnetic beads. So you can see underneath, maybe you can see, underneath this is a little thing that I had the movie, which I don't have, I have it, but I'm not showing it. We come up here and basically do microfluidics mixing. So you put reagents in there, and if you look between these two things, right, separate dots, and it will mix these things, right? So what we're interested in doing is building a compact unit that would combine the liquid handling automation with the sequencing automation with the classification technology that we've been building. So this shows, Klebsiella, Mycobacterium tuberculosis, difficile, Staph aureus, and so on, all these different strains for which we've built classifiers for antibiotics, so we can, just from the genome data alone, depending on what isolate you have, make a very accurate prediction as to whether or not it's antibiotic resistant. And this is very useful clinically, or in national security applications, or in field hospitals. So where this technology is going is Oxford Nanopore has built an even smaller version, and we're in discussions with them about how we would partner with their hardware technology with our software technology, which I'm not really telling you much about except for the fact that we've been working on it for 15 years and it's the most widely used system for annotating genomes, and to bundle some kind of AI capability with this device. Of course, you might be able to, if you have a cell phone, you might be able to use the cell phone for doing inferencing, but to make that a very low-powered kind of portable device. Now, I'm going to shift gears. I'm going to talk about a problem I'm really fascinated with. This is a joint project with the Marine Biology Lab at Woods Hole and the computational neuroscience guys at Chicago and the cephalopod biology crew at Chicago in the physics department, actually. So this cephalopods, these are octopus, cuttlefish, things like that. And they're very interesting creatures, right? They show a lot of convergent evolution with mammals, even though they're invertebrates. Their eye is almost as sophisticated as ours. It's turned the right way around. But what's fascinating about these things is that they have ability to dynamically change the patterns on their skin. And they do this in a very different way than most animals. Most animals do it through diffusion, so the cells react to hormones. In the case of cephalopods, they do it by having little structures called chromatophores that are connected directly to their nervous system. So, for example, in the top here, you can see 3 different time steps of how this animal was changing from camouflage pattern when it's resting on the ground to something that is more visible as it's getting ready to move. And it does this by directly controlling these structures in the skin. So it has chromatophores, which are basically little sacs that have pigment in them, and with a neural signal, they can open and close the sac, okay? They have iridophores, which are below that, which actually reflect light, and they can control that to create an iridescent kind of effect. And they have some other surfaces that change the surface, the texture of their skin, right? So here's a picture on the left of one of these chromatophores. Now, an octopus has about 500 million neurons, and about 500,000 of them are dedicated to managing this camouflage apparatus in their skin. This is what it looks like if you kind of zoom in on it. And what we're interested in here is the following biological problem. So octopods are extremely evolved in camouflage. They're probably the best animal for camouflage. And they control color, and they're very sensitive to the color backgrounds that they operate in. But they're also colorblind. So it's completely unknown how they manage to match the color of their skin with the color of the surface when they can't actually detect the color. So what we're doing is we're building a network structure, computational structure, that tries to learn how to do that without actually having eyes that can see color. It's a result of a bet I made with Henry Markham about evolution versus machine learning. But just to put it in some perspective, I mean, this thing has 500 million neurons. A snail, which is similar in some sense, invertebrate has about 10K neurons, so maybe we should start with snails. An insect brain has about 1 million neurons. And if you read this quote, it basically says, they have a mammalian-like anatomical organization of these higher brain functions, right? And what this is showing is that it highlights the importance of network connectivity rather than the property of single cells. So what we want to do in this project is couple a very rapid ability to construct a network model, evaluate it on the neuromorphic hardware, and loop that through a evolutionary algorithm so we can test off hypotheses between how much the system is learning from evolution, that is over generational time, and how much it's learning from local information. And if I win, Henry has to buy me a bottle of something. Okay, another project we have led by Bobby Castori, who's one of the leaders in the Brain Connectome program, And this is we have quite a large number of people involved in this. And the insight from this is that our brains are wired up in some complex fashion. This is just some cartoon from the cortex. But the way we're going to try to learn this is by taking real brains. I'll show you a picture of slicing them and processing them, scanning them, reconstructing a network through computation, doing some automated analysis, and seeing to the degree at which we can get interesting insights on brain circuits from that. So the typical sample prep, we work on all kinds of things: mice, little cephalopods, little tiny cuttlefish. I'll talk a little bit about the pygmy shrew, which is kind of interesting. But anyway, it takes about a week to extract the brain. You basically preserve it, you slice it into kind of 100-micron slices. You stain it with heavy metals, and you stick it into a high-throughput electron microscope. And what you get out of that is a whole pile, a stack of electron microscopy images, electron micrographs. You then try to recognize the membrane, so you can now trace objects in 3D. You do segmentation fusing, and you build a 3D model, and you kind of stare at it and see if it's biologically plausible. We're also doing the same thing in the advanced photon source. So this is a reconstruction of a punch out of a mouse brain done from the advanced photon source using computerized tomography. And the round things are neuronal bodies, a soma, that is big enough to see, because the resolution in the X-ray is not so big, but the red is the vasculature. And of course, what we can really do is reconstruct the vasculature. But if we superimpose in that the electron microscopy, you can, and here I'm just zooming through the slices here, the coloring corresponds between the neurons, so it's been segmented. There's a very large machine learning data set we're using for driving the segmentation, but what you get when you do that is a volume that you can reconstruct. And from that volume, you can go in and, using topological analysis, you can pull out individual neurons and reconstruct a neural map in the kind of abstract. And so this is what we're really interested in experimenting with, are these neuron maps, and defining neural circuits, and then using what amounts to the neuromorphic hardware as an accelerator to loop through experiments in structure. And I won't go through that. But the particularly interesting target here is a pygmy shrew. And the reason that this is interesting, how many of you have pygmy shrews as personal friends? Yeah, not so many. Okay, so I didn't even know about this, so I started hanging out with Bobby. He's a crazy MD, PhD, crazy man. And he said, well, the reason we have to work on pygmy shrews is they have the smallest mammalian brain. I'm like, cool. So we start reading the literature about this, and I learned one really interesting, first of all, this brain is about, it's not more than a couple of millimeters. It's really tiny. It's like a grain of sand. But these things are basically blind, so they don't have much in terms of visual processing. But they have this amazing nose with 10,000 whiskers on it. They have like the most coverage of whiskers per cubic, density of any animal. And their main diet is crickets, okay, which they hunt in the dark. And this has been a fascinating thing of how they actually are managed to hunt these crickets. And they do it purely tactily. So you can see on the right-hand side here, this 30-degree lobe is because they have all these whiskers, and the whiskers are, if you look at their brain, what we would devote to vision, they devote to processing the whisker input. And here's a timing diagram of how fast they can attack these things, and that poor cricket on the left, those yellow dots are bite marks, showing that the shrew is able to actually get its main teeth right on the part of the cricket needs to kill it. And so what we want to do here is, because this brain is very tiny, we actually can reconstruct it in a few months and build, again, a circuit simulator that would allow us to reproduce some of the experimental data. I guess I'm getting the hook here. One more, and then I'm done. But this is my favorite project. So I like to play with drones, and the project that I'm working on is one where we want to equip drones with hyperspectral imagers. Of course, this is being done for precision agriculture. But what I want to do, aside from building these response cubes, is to have an intelligent controller chip on here, and again, neuromorphic is one way to do this, that will allow me to train it on interesting sites. For example, in the Burgess Shale, train it on sites where we have known fossils of different strata, get the different spectra, and then work with my colleague Neil Shubin, who does, Neil was the one who's found the oldest tetrapod fossil up in Ellesmere Island. So there's Ellesmere Island. I put the map on here so you see where it is. It's up by Greenland. It's the kind of northernmost island in Canada. It's the 10th largest island in the world. 75,000 square miles, 150 people live there in one little place. This is what is beautiful, okay? But it's incredibly difficult to search. So the idea is to program the drone, again, interpreting the multi-hyperspectral imaging, but instead of doing what's normally done, like in precision agriculture, you fly the drones, you take the data back, you process it, batch, and so on. We want to make it dynamic so that the drone will actually, if it starts to get a signature, it will then zoom in and map sites. And the reason ultimately we want to do that is that if we wanted to, say, explore Mars and look for interesting signatures, whether it's for life or possible fossil life or remnants of Elon's car at some point, we need to be able to reliably take hyperspectral images, process it, and have a fully autonomous search. So those are the projects I'm spending, there's more, but I won't talk about, and let me just finish here. Happy to say more at the break. Okay, that's it. Done.